actually in our, actually, I think we have told the participants to join at, in about 15 minutes before the start of the talk. So I think a few more participants will be joining. So it's 5:56 in Indian Standard Time, and we have officially your talk will start from 6 p.m. So another 3 or 4 minutes.
Excellent. This was a long Friday for you. Full day of work.
Yeah, it is a great experience. It's a great experience. I have, we have organized talks, but it's kind of, it's a different experience altogether. Sitting in a room and actually talking to people instead of going to a podium and, meeting with the participants, with the speaker. It's a different kind of feel. I know everybody's going through that probably.
Yeah, definitely. It's very exciting. It's a new kind of living, meeting many people worldwide at the same time without traveling up and down. And, it's really, it's true, it's virtual, but it's like we are in the same place, and it's very unique as feeling. So this is one of the good side of this pandemic. Living with this new version of discussion, meetings, and conferences. Yeah.
It's very convenient, isn't it?
Yeah, very convenient.
I will probably miss interacting with people in person, but
yeah.
At least we can get it done like this. So it's always good, and we have to see the positives, don't we?
Yeah. And how about you, in your university or institution, students, are they allowed there in the campus?
Here they are now. I think it was just like 6 months ago, maybe less, 5 months ago, that they started bringing students in. But this year they are going back. Actually, for postgraduate studies, it will be next week, I think, beginning of October. So let's see how it works. It's scary, but have to carry on.
Yeah. How about you,
Oh,
Vic?
here they are still allowed.
Still allowed?
Yes, still allowed, but we avoid meeting with more than 50 participants. The government try to regulate that, but generally they are still allowed to come to campus and move. Everybody is moving normally, but you need to have your gel, you need to wash your hands many times, you need to wear your face mask when you are among people. Here, I'm not wearing it because I'm alone in my office, so no need it. But before getting out, then you need to reach and take your mask.
Yeah. So you have to follow all the regulations, basically, when you are in the campus.
Yes, you need to follow it. Even if you forget, there is someone to remind you that you should wear your mask or something like that.
Great. So we can start now. It's 6 p.m. So let us get going. So first of all, hello everyone. Welcome to the Champions Talk. We are organizing this event as part of championship event. First of all, I like to thank all the presenters who agreed to participate and present their research and their talk in this event. We have organized previously championship event at the Adamas University during 2017 to 20. This is the first championship event which is completely online here at Adamas University. So again, thank you all to all the presenters and all the participants who are present today, and some of the participants might be joining. So we have 3 speakers today who will be speaking. So the first speaker today is Dr. Karen Campos. She holds a BSc in biology and MSc in genetics and molecular biology, both of which were awarded in Mexico City. Later on, she moved to the United Kingdom to pursue a career in science and obtained a PhD in biochemistry at the University of Bristol in 2013. Dr. Karen has nearly 6 years of experience as a postdoc research fellow. During this time, she worked at the Institute of Cancer and Genomic Sciences at the University of Birmingham for 5 years, as well as at the Faculty of Biological Sciences at the University of Leeds. At the moment, she's in a career break due to certain family reasons. Her primary research interest has been the study of viruses that cause human diseases. It's a very interesting topic. Most of her research has been focused on the molecular biology of human papillomaviruses, or HPV in short. HPV are the causative agents of benign and malignant diseases, including cervical, oropharyngeal, and other types of cancer. As a postdoc research fellow, she undertook different research projects that aim to understand the mechanisms that HPV use to persist and replicate in their host using primary keratinocyte-based models of HPV infection. Our talk today is short stories about human papillomavirus. With that, we'd like to welcome Dr. Karen for our talk.
Thank you very much, Dr. Arindam. I want to thank you for inviting me to give this talk today, for organizing the whole event and putting us together. I'm gonna share the screen now. Let's just start with the presentation. And well, my talk today is gonna be some short stories that include some of the research that I performed as during my career in the UK as a PhD student and as a postdoctoral researcher. I just decided to just give you some highlights. I'm not gonna go into much detail about each of them, but I wanted to mention some of those because I wanted you to just leave this presentation knowing a little bit more of this virus. So papillomaviruses are small DNA viruses that infect a great variety of animal species. And there is loads of information about bovine papillomaviruses because of their economical importance. However, our main focus is to study human papillomaviruses because of their medical and also economical importance. So human papillomaviruses are small DNA tumor viruses. So basically they have just one, sorry, let me just put a pointer here. Yeah, it's only one molecule of DNA, circular DNA. And these viruses are non-enveloped. Basically, they are protected by a capsid, which is formed of 2 main proteins. I'm gonna explain this to you later. This HPV genome encodes, well, it's about 8,000 base pairs, and it encodes 8 open reading frames. And these 8 open reading frames encode early and late expression proteins. So basically these proteins are expressed in different times. Oh, sorry. Shall I carry on? Sorry, I don't know what happened. Okay, so so these proteins are expressed early and late depending on the viral life cycle, which I'm going to mention in a minute. And there is also a long control region, sorry, which is a regulatory sequence that contains binding sites for several cellular or also viral proteins. So to date, there are more than 150 HPV types already identified, and those that infect the mucosal epithelium can be divided into groups. Low-risk types, such as HPV 6 and 11, cause warts in the genital tract. They are benign lesions. However, high-risk types, such as HPV 16 and 18, cause cervical cancer. And I just put an asterisk here because it's not the only type of cancer that these viruses cause. And I'm going to mention this in a second. But I wanted to draw to your attention that actually the link between human papillomaviruses and cancer was only discovered very recently by Professor Harald Sorhausen. He won the Nobel Prize in Medicine in 2008. And actually he was awarded this because he discovered the relationship between these viruses and cervical disease. So what he and his group did, well, they isolated and partially characterized HPV DNA that came from a cervical cancer biopsy. And then they took this HPV DNA and then compared and evaluated the presence of it in additional cancer biopsies, as well in cell lines that were derived from cervical cancer. And here you can see that there is a high percentage of total positive biopsies for HPV 18. So HPV not only causes cancer in the cervix. Here you can see that there are different types of cancer that can be present either in males or females. And actually the size of the circle indicates how many cases in the UK. Obviously this can vary worldwide, but I don't think that you will see many differences. Obviously the numbers would change a little bit, but the important message is that HPV not only causes cancer in women, so it's really important that men are also protected. And how can we protect men and women against HPV? Well, fortunately, now we have vaccines available. So in the case of, we have Cervarix, which is a bivalent vaccine. It protects against two high-risk HPV types, 16 and 18. Gardasil, which is a quadrivalent vaccine. It protects against 9 types of HPV. The quadrivalent protects against 4, which is 2 types, HPV 6 and 11, and high-risk types 16 and 18. And there is also another more recent vaccine that is an updated version of this one. Let me just tell you quickly that Gardasil has been the HPV vaccine that has been used in the NHS vaccination program since 2012. This vaccine is applied to girls and boys aged 12 and 13 years old. And apparently there are plans to change this vaccine and update the program to apply Gardasil 9. And actually this new vaccine, it protects against more HPV types. I would like to mention that HPV 16 and 18 are the cause of most cervical cancers in the UK. They account to more than 80% of the cases. However, high-risk types 31, 33, 45, 52, and 58, which are also high-risk types, can cause an additional 15% of cancer types. So it's important to know that these vaccines not only protect against these high-risk types that cause cancer, but also against types that cause genital warts. So basically we are protecting here people from getting malignant diseases, but also other problematic diseases that can cause economic impact around the world. So these vaccines, I don't know why I cannot go forward. Okay, there you go, sorry. So these vaccines were developed by Dr. Ian Fraser and Jian Su at the University of Queensland, Australia, and they use recombinant L1 proteins. So the viral capsid is formed of these proteins called L1. They form a pentamer themselves. There's another protein called L2 that goes in here, but actually the vaccine only contains the L1 recombinant protein. And actually once the viral particles, they are called viral particles because virus-like particles, they look like viral particles, but they are not viral particles. So you have virus particles that contain the whole HPV genome, all of the HPV DNA and complete proteins. However, the viral-like particles are the ones that are used for the vaccine, and they only contain this recombinant L1 protein. So there is so much that you can read about this vaccine. I suggest that you have a look at these references that I add here if you want to get any extra information or further reading about this topic. Well, the HPV life cycle is quite complex. It depends on the differentiation program of the keratinocytes, the epithelial keratinocytes, which are the host cells for this virus. The virus enters the epithelia through small lesions, then it infects cells that are in the basal layer. And in this region, there is some replication that occurs in order to achieve the maintenance of the viral genome. So for this to happen, early viral proteins have to be expressed. The viral proteins E1, E2, and E4, they are important for viral replication. I'm going to talk to you a bit more about E2 in a minute. But also there is more known about the E6 and E7 proteins. With E5, all of them are considered oncoproteins because they actually can interfere with the cell cycle. Actually, E6 and E7 have the ability to bind and degrade P53 and RB, and that's the way that they basically abrogate the cellular checkpoints and allow the cells to proliferate. The cells that are in the upper layers of the epithelium should not proliferate once they are differentiated. So it's actually these proteins that allow these cell cycle controls to be stopped. So there is an increase of viral DNA amplification. In the upper layers of the epithelium occurs the release of the mature virions, and then these virions have the ability to go and infect more cells. So most of my research was focused on the HPV2 protein. This protein is a small protein that can dimerize, so it works in dimers. It has a transcription or replication domain. So this N-terminus domain is known to be important for interactions between E2 and other cellular proteins. There's a hinge region that gives this protein some flexibility. And why is this flexibility needed? Well, because E2 is a transcription factor. So it has the ability to bind to DNA. This C-terminus domain is the DNA binding domain. And the functions of the E2 protein are viral DNA replication. It needs other viral proteins and cellular proteins to achieve this. Transcriptional regulation. So this is the only transcription factor that the HPV genome encodes. And it also is important for viral genome segregation and maintenance. So during my PhD, what I wanted to evaluate, basically I have a project in which I have E2 proteins from high risk and low risk settings, basically HPV6, which is a low risk, and HPV16, high risk. And what I did first, this is only a little bit of my project, one of my projects as a PhD student. So I measured the E2 DNA binding activity of these two proteins. So something that I mentioned before is E2 has these two domains. However, the whole protein has not been purified as a full-length protein. So during my PhD studies, I only use this DNA C-terminus domain. It's only the C-terminus part of the protein that I'm gonna show you the results for. So this protein binds to the DNA, but it has to recognize a specific site. This is called an E2 consensus binding site. These sites are in the viral long control region, and they could have either a high presence of AT or CG nucleotides. If I talk to you about a DNA that has loads of AT nucleotides, that means that that DNA is flexible. If it has more CG residues, it's less flexible. So if you think about a protein, the protein, if it binds to a DNA that is very inflexible, the protein has to be flexible itself. However, if you have a DNA that is very flexible, then maybe the protein doesn't have to be as flexible. I don't know if that makes sense. I hope I'm making sense here. So what I found was that this is an EMSA, electrophoretic mobility shift assay, in which I incubated radio-labeled DNA and purified E2 protein, the DBD domain. And here, if you look at here, this is the complex of E2 bound to DNA. And you can see that there is less protein required to bind to the DNA when you compare it to this other protein. This is a low risk type and then a high risk type. So what this tells us is that this low risk type protein has a very high affinity for its DNA. This is an 80 Ridge spacer. So that means that this protein is more rigid than the 16E2 type. Why is this important? Because there are differences between the proteins that are expressed from low risk and high risk types. And these differences might be very important for how the virus behaves in the body and why a virus is causing a malignant disease, or why is it only causing a benign lesion. So just quickly, what we wanted to know, what is this protein doing? We basically look at the structure of this protein and we saw that there are 2 leucines at the end of this protein that are not present in the high risk type. So basically we remove those leucines by site-directed mutagenesis, and then we perform some studies. So basically you have the wild type protein and the truncated protein here. And you can see that the protein that does not have the leucines can bind much better. You can see here compared to here, you can see some binding in this line. This is better than the wild type protein. This is an 80 spacer, basically a more flexible DNA, but then when you look at the more rigid DNA, there is practically no binding with the 62 wild type protein, but we could see some binding with the truncated mutant. This is specific DNA. I also used a DNA that is non-specific, and you can see that there is more binding of the E2 protein to this DNA. So basically all of these are E2 DNA complexes. So what these studies showed was that removing those leucines increased the affinity for specific and non-specific DNA. So this is just part of this study. When I started working as a postdoctoral researcher, I moved completely out of the biochemical side of it. I used more cell-based research. I worked with a protein called RAD50 interacting protein 1. This protein has the ability to modulate the subcellular trafficking between the endoplasmic reticulum and the Golgi apparatus, and also between the Golgi apparatus and endosomes. This protein is a cellular protein. And what I did was I used C33A cells, which come from it's basically cells derived from cervical cancer, but they do not contain HPV. And I performed immunoprecipitation assays in which basically I used an antibody that recognized endogenous RIN1, which is this protein, then removed everything else that didn't really bind to this complex, and then performed Western blotting. And you can see that HPV16E2 binds and immunoprecipitates with this cellular protein. So I characterize this binding in more detail. You will probably, if you're interested, you can see this in the paper itself. But something really interesting is that when I looked at immunofluorescence, you can see that the E2 protein localizes to the nucleus of the cells. And RIN1 also does that. So when E2 and RIN1 are expressed together, you can see that they co-localize in the nucleus. And as I mentioned to you before, E2, E1, and the origin of replication are necessary for the replication of the viral genome. So it was really interesting to see that RIN1 co-localized with these proteins that are viral proteins, and that actually these proteins are necessary for viral replication. So we wondered if actually what E2 was doing was recruiting RIN1 to this origin of viral replication to aid actually that function. And yes, that was the answer. This is just a Western blot to show you the expression of all these proteins. This is a transient replication assay in which, you have all the controls here. And this is what you should look at. Basically, when you have the HPV E1 and E2 proteins in the presence of the origin of replication, you get replication. And as you add increasing amounts of RIN1 protein, then this replication is enhanced. So that was my first postdoc. When I started my second long postdoc, the project was completely different. I'm going to mention to you a little bit more about the details of who I work with in these projects. Dr. Joe Parrish was my mentor and boss, and an excellent person in the HPV field. I will mention to you later on about this. But basically, the project that I joined afterwards was to work with this transcription factor called CCCP-C binding factor, or CTCF. This protein is essential for genome-wide organization and remodeling of chromatin. It's a hot topic at the moment. I'm just gonna be as brief as I can with this project, which is very complex. But I'm just gonna give you a couple of highlights. This protein is an insulator of chromatin domains. It facilitates the communication between DNA sequences, for example, between enhancers and promoters. It directs nucleosome positioning, and it mediates long-range chromosomal interactions through DNA loop formation. So when I think you remember this from the beginning of my presentation, I told you that lots of proteins bind to the LCR. However, there are many other proteins which can also bind to other parts of the HPV genome. And actually, there are CTCF binding sites within the HPV genome. This was observed by someone, Christian Parrish, who worked with Joe Parrish. And what they found is that there were these binding sites. And actually, something really interesting is that here you can see CTCF binding sites in blue. And what you can see is that there is a conserved CTCF binding site within the E2 open reading frame of high-risk viruses, but not in low-risk viruses. So this site is only conserved in high-risk viruses. So there is a big difference here. Most of our research was focused on HPV-18. So what we basically carried on working was with this genome. This is a model in which basically we can study HPV biology. This is performed at the University of Birmingham. Dr. Sally Roberts and Joe Parrish are excellent at doing this. There are some other people in the States and also in the UK who work with these cells. And basically, we work with primary human foreskin keratinocytes that are transfected with the HPV genome and also another plasmid to give some neomycin resistance. So you get a mixed population that contains these viruses. It has to go through antibiotic selection, which is given by this plasmid. We grow these cells for an extended culture of 8 to 12 weeks, so we can actually establish HPV positive cell lines that contain episomal HPV genomes. This work is really laborious and it looks very straightforward, but it is hard work, and it is all worth it because we can actually use all these cells to be grown in monolayer, which are undifferentiated, or we can differentiate them in methyl cellulose. So basically, we can actually recapitulate the viral life cycle in these cells in the lab, which is amazing because obviously it's an epithelia. It's not easy to do this. So just to conclude, and my last slide wants to show you that actually this CTCF binding protein that I show you that bound to the E2ORF in silico and in vitro also binds in vivo. So I perform chromatin immunoprecipitation. This is also performed, well, this particular experiment was performed by Dr. Aisha Pentland in Joe Parrish's lab. And what this happens is this experiment looks at the proteins that are bound to a specific sequence of DNA. This DNA is cut, so we have smaller pieces. Then we have an antibody that is specific for this protein. In this case, it was a CTCF antibody, and then we remove this crosslink. So the protein no longer stays there, and then we have just fragments of DNA that we can use as template for qPCR. So what you are looking here is the binding of CTCF to all of the regions of the HPV genome. So you will see that CTCF binds to several regions, but actually there is a very strong binding to this E2 open reading frame. And this was basically, this is our experiment in a genome that does not have the CTCF binding site. Basically, the CTCF binding site was mutated. You don't see this binding. So it proves that this binding to CTCF is real. So this is basically important. Why? Because apparently what we observed was that CTCF, when you don't see the binding of CTCF, E6 and E7 transcripts and E6 and E7 proteins are expressed highly. So basically CTCF is regulating the expression of these HBV oncogenes. And I won't have time to tell you this later on, but in the paper you can read more about it. This shows that CTCF regulation is differentiation dependent. And why is this so important? Because CTCF function is altered in malignant disease progression and carcinogenesis caused by HBV. So I hope that this presentation gave you a little bit of insight about what are the differences between high risk, low risk viruses, differences about different projects that you can work on if you're interested in microbiology. I want to thank my mentors, supervisors, Professor Kevin Gaston. I was his PhD student in the University of Bristol, and Professor Joe Parrish and Dr. Sally Roberts, both other groups. I work with Professor Parrish at the University of Birmingham. All these people have given funding to these projects. And most of all, I would like to thank the Microbiology Society, not only for allowing us to have this kind of events, but also because they have helped me immensely throughout my career as a researcher. So thank you. I think that's on my part. Let me just stop this, and that's it. There you go.
Yeah, that's a great talk. We have some time for questions, so please go ahead. You can unmute yourself. You can raise your hand feature, or you can just write in the chat if you have any specific question.
Sorry, it might be a lot of information going on. It was, there you go. Excellent. Okay. I'm going to read this loud so other people can see. So thank you. I've seen. Is biopsy the only way to diagnose the infection to HPV? Does the vaccine? Okay. Actually, these days you can diagnose maybe, yeah, to know in more detail about what's the status of the tissue, or if there's something malignant being observed. But what I was gonna say is that actually the screening programs these days, pap smears, you can actually give your sample for pap smear, and people can get the results back knowing that if you have HPV or not HPV. You can actually perform that with qPCR in a very effective way. So probably the beginning of the of assessing if someone has got HPV, that can be done without a biopsy, but probably more in the future to know more about if there is cancer, or if there is a lesion that is only benign, then that probably has to be done by a biopsy. And then, does the vaccine have the same rate of accuracy for all HPV types? Well, so let's say these vaccines, the non-valent vaccine that covers for 7 high-risk types, that should be pretty effective for all of them. However, outside of those 7 types that are not included in the vaccine, that might basically, people can get a little bit of protection by cross-reaction. So I'm going to answer this with a very actual subject. You have this COVID vaccine that can protect against some variants and some we don't know. So basically, it depends. It depends on the type of HPV. However, I would like to mention that because all of these types that are actually being the ones that the vaccine is directed to are the most common ones, that they account for many of the infection numbers, that would give a really, really good accuracy and a really good protection against disease due to HPV. How do you analyze the protein DNA binding? Yeah, so for protein DNA binding, after doing MSAS, in my particular experience, what I had to just analyze it by densitometry. I had to just quantify it. There are other ways, as you mentioned. Maybe you can get all of this Fred, Brett, all of that. It can give you more idea of the activity of the protein. So basically, yeah, back when I studied my PhD, that was kind of like a little bit more basic. But now you have many other ways in which you can actually analyze your information, your binding, and get more of the information that you get, which can give you activity, as you mentioned, and that's it. So, okay, thank you very much for your questions. Professor Alarcón, how does CTCF regulate the expression of E6 and E7? Yes, so it does. So when CTCF binds to this, it's a very complex subject, that one. So CTCF has the ability to form loops with any DNA. So that E2 binding site, no, the site of CTCF that is in the E2 open reading frame is about 3 kilobases downstream the LCR. So CTCF has the ability to bind to other cellular proteins. In this particular research project, what we found is that CTCF binds to YY1, which is a transcription factor that has binding sites in the long control region. So CTCF binds with YY1, this forms a loop. And this loop, when it is formed, it's repressing the expression of these two proteins because it stops the activity of that promoter, the early promoter, that is where these proteins express from. However, when this loop is disrupted either by absence of binding CTCF or YY1 or knockdown of YY1 or CTCF, then the loop doesn't form anymore, and then basically everything becomes available, so the transcription of the viral genes can occur. So thank you very much for your question. Currently, what are the types that have been used in vaccines because of the distribution of the types? Well, yeah, actually, I worked with HPV when I was in my master's degree in Mexico, and I worked with HPV 16, and in between, let's say HPV 16, that's a type, but there are also subtypes, and these subtypes are more often found. There are some subtypes that are more often found in one region, some other types are more often in another region. Obviously, worldwide, it's completely complicated. However, the advantage that HPV has is that it's a DNA virus, so it mutates less quickly as an RNA virus. So it has been possible to protect people who have been vaccinated. Obviously, it has been, how many years? 15 years? No, 10, 12 years, more or less. So the studies are happening. There is a protection. People who have been vaccinated long ago, obviously all of this forms part of a trial because of how long it takes to, see the efficacy and long-lasting effects of protection of a vaccine. But the effect has been at least 10 years. It has happened. It has protected against HPV types. People who have been vaccinated, they seem to be protected. So definitely, it's important to get vaccinated. Let me see, sorry. Distribution type, thank you very much. Thank you for your comments. How is AT more, okay, so the composition, how is AT more flexible than CG? That's the question. So the nucleotides, it depends on the structure of the DNA. So because of the formation of these nucleotides, how they have their bases, the sugars, everything, how it's basically positioned, you can see that G and C, guanidine and cytosine, they have more rigid structure per se. So if you have loads of them, loads of those nucleotides together being quite rigid, that's going to create that the whole DNA molecule is going to be more rigid, whereas if you have adenine and thymidine in the DNA, then there will be more space in between them. So basically it's mechanistically, it's kind of part of the structure of the DNA. Oh, that's very kind of you. Thank you very much for your comments. I greatly appreciate it. I really hope that my speed was not too much. I wanted to tell you so much about it and not take that long, but I hope that you can leave today knowing a little bit more about this virus that I have worked with for so long. I think that's for the questions. Also, I forgot to say, if you want to have more information, access to the papers that I mentioned to you today, you can always find me on social media or send me an email, and I would be happy to provide you with more information about it.
Is there any animal model for testing the vaccines?
Some of the research, there are many groups that have worked with mice. I think that's as far as it has gone. With the vaccines, would you mean like where was the vaccine tested and all of that information, or if there are being more studies about? Yeah. I think that basically most of the studies are in mice.
Nice.
Yeah, and actually there are mice that they are very famous. They are like knockout E6 and E7 mice. So basically, you just work with them and then compare them to wild type mouse, and it's just very quickly. There's loads in Mexico in particular. There's a group in the States who uses mouse models intensively, and obviously for the vaccines, probably they use this model to check just before giving it to people. I don't know.
Am I allowed to go live?
Oh, thank you, Dr. Ramirez. Oh, thank you. Thank you very much for all your kind comments.
Greeting, Doc. I have one question, Dr. Campos.
Give me a second. Yeah, can HPV target mesenchymal tissue? I could say, I'm not sure, but I don't think so. The way that HPV, or at least not that I know that having any reports about it. So the keratinocytes, they have receptors such as any other cell in the body, and some cells are more prone to be infected by certain viruses depending on the receptor that they use to enter the cell. So HPV is very specific for certain type of epithelial cells. And I'm saying epithelial cells because, as I mentioned before, HPV can cause other types of cancer. Before, we didn't know that most head and neck cancers were also associated with HPV infections, such as mouth, throat, oropharyngeal cancers. And obviously it depends on what are the cells that are forming that particular tissue. So I would probably say, I think that it's more specific infection towards keratinocytes, those particular cells. Why? Because of the receptors. I don't know if I am missing any kind of current information, but I think that would be my answer.
Yes, Dr. Campos, I have one question to ask.
Yes.
Thanks first of all for your presentation. My question is, a lot of things has been done on HPV, by the way, from vaccine and to the search for treatment. So today we are celebrating International Microorganism Day. And I'm sure there are many young scientists who would like to take something from your research and be able to maybe follow you and follow your career. So what can we have as a current trend in papillomavirus research for future scientists?
Yeah. So I think lots of research groups that are actually, well, at this time working with papillomaviruses are doing, first of all, everyone's trying to move to sequencing techniques because it's much easier. You get so much information from next generation sequencing, RNA sequencing, chip sequencing. So basically, that is what is hot at the moment on papillomaviruses. Why? Because then you can actually see what is happening in terms of expression and association to different kind of cellular proteins. Let's say in the past, you have, everyone was spot on, E6 binds to P53, P53 was hot on, let's just go for P53, And right now it's more about what is exactly what's going on, what kind of cellular proteins are being targeted by these viruses, how is possible for the virus to persist, and what makes, basically, something I forgot in my talk is, not because someone has been infected by a high risk type, that means that that person is going to get cancer. Cancer is a multifactorial disease. It not only depends on HPV infection. However, people who already have got cervical cancer, 99% of those people are HPV positive for a high risk type. And I'm saying this because we want to know what is making this difference between people who can get infected and can actually clear the disease, clear the infection, and what happens to people who can't, and what happens within the cells, like what are the proteins that are protecting the body against it? Are they tumor suppressor proteins? Is it a combination of all of them? Is it a variety of arrays of, like proteins that are involved in a particular pathway or immune system? So it is very complex, but basically this is what is happening at the moment. It's just, and why, if we have a prophylactic vaccine, we want to carry on studying papillomaviruses? Well, it's just around the world. This happens in people who have the access, like the access to the vaccines. But many countries, like my country, even though people are getting vaccinated, lots of other people are, suffer from cancer. So how can we actually treat them? How can we give those people a better quality of life? So it is important for everyone to carry on, not thinking that the vaccine is going to solve a problem. And I think that applies to every kind of virus or disease. I'm sorry, did I answer your question, Dr. Arindam?
Yes, you did. Thank you.
Thank you.
Is there any other question? Do you factor individuals' genotype before? Do you consider the genotype of individuals before giving the vaccine?
I don't think that people are assessing that at the moment, really. I think that probably our current situation is an example of what happens, like probably it would take a bit of time to assess who might be eligible for a vaccine, and that might really take, that time might be the time that that person has lost its chance to get the protection. I want to say that that vaccine is very safe. It's been a long time now. So at least we are seeing that there is protection. Things are going well. It is not given to people who are already, like over 30s or 20 something, because most people will encounter a HPV infection throughout their lifetime. So basically, healthcare systems think that that is pointless. However, I would say that for young adults, it would be worth to take that, to take all the chances that you can have to protect yourselves from a disease.
I have just a general question because there are a lot of students. So what do you think, students who are pursuing research as a career, what are your few tips to them, this budding generation?
About them being carriers, you mean?
Yeah.
Okay.
Yeah.
So obviously, HPV is associated with sexual practices. So it's a difficult subject because as long as someone, if someone has multiple sexual partners, that complicates things. As I mentioned in the past, this vaccine was given to girls. However, boys, even though they cannot get cervical cancer, they can get other types of cancer. Now we know it, but they can also pass it easily without even noticing that they have a problem, whereas a girl might just notice it faster because it's cervical cancer and the disease and all, like discomfort and many things that can be observed much earlier. So probably I would say that with any kind of virus, I think this applies to everything. I think that we all have to be very careful. We cannot live worrying about which virus we might get. Unfortunately, we have to live like this. We are evolving, viruses evolve with us, they evolve faster than us. And we just need to make sure that we know how to keep ourselves safe. So the best practices we have, not only sexual practices, but hygienical practices, with hygiene. You can see what's happening with COVID and many other viruses. As long as we are careful and we are clean and we can actually limit the kind of exposure that we have to unhealthy or any kind of dirty environment, that would be good. I just think that probably for the next generations, it's very important to learn and read about many viruses, because you can learn so much and you will find that some of them behave similarly. So you would probably learn from one virus and then learn without knowing about other viruses. So I think it's really important that all the generations keep, not only youngsters, but all of us. I keep learning every single day, and there is so much that I don't know.
Great. So my question was actually much more generic for students who have not started a research career. So, what kind of, it's a general question actually, it's not specific about the awareness, because there are a lot of students, that's why I asked this question.
I can do that as well. I think that's going to be listen. Okay, I think that students, use your time wisely. I think it's really important that the younger you are, the younger you can start. If you already know what you like, if you are really interested in science, get involved in activities that are associated with science. It doesn't matter that it's an activity that might be for you like, oh, it's just this little event. No, everything is big, everything counts, everything gets you closer. So approach senior people, researchers, professors, everyone that can basically learn as much as you can. I think that is the best thing that, or what I would say to myself, I still think that I could have done so much with my time when I was young. I think that would be and read a lot. I think that is the best way of getting a better career. It doesn't matter what career they choose.
Excellent tips. Thank you so much.
And I gave them a bit of advice from other things.
I think we do not have any further question probably, and those participants who would like to might think some question, maybe after some time they can just send us an email. We'll definitely get back to you.
I just want to thank everyone for attending this presentation, for your amazing questions. Thank you for each of you for that, and for you, obviously, who are here, everyone attending the presentation, and for you, Dr. Arindam.
Thank you so much. Thank you for actually agreeing and presenting and also interacting with the audience and answering so many questions. So your presentation and your answering the question is almost like 50-50, the time you spent. So it's very good. It's very good.
Thank you. Sorry, I hope that it's not like it wasn't too long, but I hope that it's useful.
It's great. Thank you so much.
Thank you. Enjoy the other presentations and talks, and enjoy the
Sure.
day.
Thanks. We'll take a maybe a break for 10 minutes, and then we'll start the next talk, because students, everybody's listening. So maybe a 10 minutes break, then we'll start with Dr. Victorian's talk. Yeah. Hello, everyone. We are going to start with the second talk. Which will be presented by Dr. Victorian. Yeah, let me introduce Dr. Victorian. Dr. Victorian is currently working as senior lecturer at the Polytechnic School of Abomey-Calavi at the University of Abomey-Calavi, Benin. Dr. Vic is involved in the fight against antimicrobial resistance in Africa. And as a scientist, his interests are linked to molecular microbiology, hygiene, as well as public health. Dr. Vic is passionate of medicinal plants that can treat infectious disease, and he does his best to impact Africa's economic development. Dr. Vic received his PhD in environmental and health science in 2013, and he completed his PhD and in 2013, and after a MSc degree in biomedical sciences in 2011. Before that, he received his bachelor in biomedical sciences in 2008. Dr. Vic did all his studies at the University of Abomey-Calavi in Benin, with many scientific visits abroad. Dr. Vic teaches microbiology and research methodology modules at undergraduate and postgraduate levels in two of the biggest universities from his country. He visited as guest lecturer in many countries such as Lebanon, Mauritius, and Togo. He has multiple collaboration. One of them is with the World Academy of Sciences. And with this collaboration, he initiated the establishment of a MSc program in molecular and medical microbiology. This program was set up to prepare some well-trained specialists in microbiology. Those specialists was very useful for Benin during Lassa fever pandemic and the recent COVID-19 outbreak as well. Dr. Vic collaborated with more than 150 researchers from 80 different research institutions around the world. Dr. Vic is a founding member of the research unit in Applied Microbiology and Pharmacology of Natural Substance, Research Laboratory in Applied Biology, Polytechnic School of Abomey-Calavi at the University of Abomey-Calavi. Annually, more than 150 young scholars are impacted by his lab's activities. His academic career has been regularly enriched by participation in international workshops, symposia like this, and his visits abroad, as well as through funded trainings by various international scientific institutions. From 2015 till date, Dr. Vic has won 14 competitive research grants as principal investigator or local coordinator, as collaborator, and he has also won more than 35 travel grants. His research outputs include 6 PhD and 25 MSc graduates. He published more than 136 peer-reviewed journal articles, 75 conference abstracts, and 22 popular articles. Dr. Vic's talk today is Challenges of AMR in West Africa, State of the Art from Beninese Researchers and How to Change the Situation. With that, we'd like to welcome Dr. Vic for his talk today.
Thank you very much, Professor Arindam. Thank you for this introduction. I will share my screen. 1 minute. Okay. Good. Do you see my screen?
Yes, we do.
Thank you very much. So I think I have been so well introduced by Prof. Arindam. I'm Victorien Douillon, senior lecturer in microbiology in Benin. And my talk will be about the challenges of antimicrobial resistance in West Africa, the state of art from some Beninese researcher, and how we could improve the situation. The outline I will follow is the following. I will present how the antimicrobial resistance is a global health challenge. And then I will talk about the situation and the challenges of AMR in West Africa. Thirdly, I will talk about the research about AMR in Benin. It will be mainly the experiences from my research team and the collaborators also. And I will finish this talk by suggesting how to improve the situation of AMR in West Africa in general and in Benin with specificity. So AMR as a global health challenge, antimicrobials at the beginning of the history of microbiology. Since that time till now, antimicrobials really help in the management of infectious diseases, but also in the reduction of mortality. We used to refer to those antimicrobials to treat diseases, to cure many diseases. So finally, it leads to the reduction of mortality. But we found that a new word came up, AMR, the antimicrobial resistance. And the antimicrobial resistance will occur when bacteria or parasites or viruses or fungi will change to protect themselves from the effects of antimicrobial drugs designed to destroy them. So that's why when we talk about AMR, it's not only antibiotics, because many times people, when they hear AMR, they just think about antibiotics. No, we have also the antivirals, the antifungals, and the antiparasitics, and all that interfere in what we call the antimicrobial resistance. This antimicrobial resistance is increasing global threat to human health. We have a numerous number of people dying from infection caused by bacteria resistant to antimicrobial each year. And from a report, especially ONE's reports, this could lead to 10 million deaths annually by 2050. So this is a huge situation, and that's led the World Health Organization to think that AMR is a global health and development threat. AMR is one of the top 10 global public health threats facing humanity. That's why we suggest, because today is the International Day for Microorganisms, we need to handle the antibiotics, the antivirals, the antifungals, and the antiparasitics with care. It's not some sweeties. We need to take them when we are sure that the medical doctor or the lab technician find the microorganism responsible of the disease and know his antibiotic pattern or antimicrobial pattern. Thank you. So how the antibiotic resistance spread? This picture show many examples of how antibiotic resistance can spread. You see, here we have the antimicrobials, and the animals get the antibiotics, and then they develop resistant bacteria in their guts. Here we have George, is a human, it could be me or you. You see, we get the antibiotics, and also we develop resistant bacteria in our guts. And when George will stay at home, he will then spread the resistant bacteria in the general community. George will go to hospital or nursing home or other inpatient care facility to get care. And there, he will also spread the germs, the microorganisms, the resistance bacteria. He will spread all this among healthcare providers because they don't clean their hands, they are working with patients, they are treating them, so they will get the resistant bacteria. And also you have the patient that will go home. And when they go back home with the resistance germs, and the cycle can continue. From the other way, the fertilizer or water containing animal feces and then the drug resistance bacteria will be used on food crops. And when it's used on food crops, then it will transfer all the resistance bacteria into the crops. And when the crops will be eaten, human will also get the resistance bacteria. So from this picture, you can see that the AMR is not only one sector problem. It's a very big problem that needs a new approach that we call One Health approach. So it's a problem for medical doctors, it's a problem for environment, it's a problem for agriculture. So every one of those sectors need to be together so that we can understand the interconnection between human, animals, and environment so that we can stop the spread of resistance pathogen. What is the situation and the challenges of AMR in West Africa? In West Africa, especially, significant resistance was reported for several bacteria in hospital or in the community. And West Africa is specifically vulnerable to the consequences of rapid AMR. Why? Because of the poverty, because of the inadequate healthcare and sanitation infrastructure, because of the poor nutrition, and also because of adverse climatic factors. Some challenges are related to education, because in West Africa, there are not enough educated experts to tackle the AMR challenge. Another challenge is related to the quality of data, because we have a poor quality of AMR-related scientific data. We have weakness in antimicrobial susceptibility testing in many countries. And even in Benin, you know, you can have many hospitals, you have many labs in those hospitals. And when you look at the antimicrobial susceptibility testing, then you can find that even the antibiotics used are not the same. So if the antibiotics used are not the same, how can we have a national situation to take decision? That is a big challenge in my own country. We have another challenge related to the data collection. Reports show that the absence of a national surveillance center that can collect, analyze data on AMR in real time. And finally, the lack of surveillance, because laboratory capacities and research infrastructure are under-resourced. The surveillance of drug resistance is not carried in many countries in Africa. And there is also a scarcity of accurate and reliable data on AMR in general, and especially on antibiotic-resistant bacteria in particular. So what about the research regarding AMR in Benin? Benin is located in West Africa. You know, it's a small country. This is my country, between the equator and the Tropic of Cancer. So this is our national colors, red, green, and yellow. And from the Ministry of Health, National Health Security Action Plan, since 2019, we prioritize tackling AMR using a One Health approach, as I said, because it's not only a problem for medical field, it's also a problem for environmental field and also agriculture. And everyone sit down together to define some strategies to overcome AMR in the country. Then we come to my lab, the Research Unit in Applied Microbiology and Pharmacology of Natural Substances, which aims to be a national unit of excellence where many scientists can work together to find solutions for health and environmental problems with major importance in Benin, Africa, and the world. So in this lab, in my lab, we work very much on AMR. We work daily on isolation of pathogenic microbes from different matrices: water, soil, hospital, environment, communities. Many places are covered by our work. We used to do some phenotypic and molecular characterization. We look for resistance genes from classical PCR or qPCR. We also look for virulence genes. And another term in our lab is the work on alternatives to antibiotics. And there, we consider medicinal plants, phages, probiotics as a good response to overcome this situation. And finally, we used to do a lot of capacity building in the field of AMR. Here we have some data about work we are doing since 3 years. We went to a hospital and we collect the environmental sample from this hospital, the catheter samples, and we found some resistance genes. And that was really interesting as result, because we found some genes that can easily be transferred to patients when they will get back home. And when those patients will come back home, they will then transfer it to their relatives and the problem will stay unsolved. We also work on chicken meat, where we found some strain of Campylobacter, very resistant. We collect some environmental, food, and kitchen sample to look for BLSE, ESBL. Also, we look for some virulence genes, and also with urine and cervicovaginal secretion in pregnant women, we detect some big resistance genes, and all these allow us to show at least the situation before holding some other grants to solve it. I will come back to that. So our lab was involved in, is still involved in many international projects about AMR. At this moment, we are conducting a 3 years project to strengthen expertise and bioinformatics to fight against antimicrobial resistance in West Africa. And this project has been funded by Erasmus+, this project from European Union. And it consists essentially on capacity building, antimicrobial resistance, and bioinformatics. We are also working with the Ministry of Foreign Affairs from Finland. And we are working to improve the capacities of lab technicians in Benin. So how to perform this antimicrobial resistance. I lost my screen. Sorry for that. 1 minute. I don't know what happened, but I lost my screen. Okay, good. It's fine. Sorry for this interruption. So with this project, we are trying to capacitate the lab technician and show them how to do the test properly, and also the detection of bloodstream infection. And another project we are doing is related to water quality, how to provide and time antibiotic resistance genes and antibiotic chemical in drinking water for quality life. So in general, we work on approximately 3,090 something samples of animals and human, and we isolate to date in our fridge, our freezer. We have 900 something bacterial strain, and the most steady bacterial strain we have were gram positive and gram negative. Now, how to improve the situation of AMR in West Africa, in Benin? The first one, the first pillar is the capacity building, because we need to work to train a new generation of researchers able to detect and monitor AMR. We are working on that, but we need to have more and more scientists able to monitor AMR. We also need to build some reference centers for the collection, the analysis, and the processing of AMR data. We need to organize some awareness activities for proper antibiotic use. We need to talk about, to show people that antibiotic is not useful when you have headache, for example. When you have flu, it's not useful, so you should not run and take some antibiotics if you don't have the evidence that a bacteria is worrying you. And we also need some AMR-focused research laboratory to technically give the strength to those laboratories in the surveillance and the conduct of AMR resistance work. Another pillar could be the alternative to antibiotics. Today, the phages are good solution. Medicinal plants, we have enough, medicinal plants. So it's good to focus on that. And all those pillars should be set up, taking into account One Health approach, because we need to raise awareness about the One Health approach definitively. We need to work together among African scientists and worldwide scientists. And for that, we had one experience with some colleagues where we put together a range of methods from microbiology, clinical medicine, sociology, and we produce new knowledge about how AMR genes can spread, especially in poor West Africa, because we were with Benin, Burkina Faso, Mali, Ghana, where local capacity to address AMR is limited. And then we identify how the spread of AMR could be changed. This will also need the acquisition of specialized equipment as MALDI-TOF or sequencer, but all this could be possible through big collaborative grants only. So you are very welcome to join, to fill the national gap related to AMR, to consolidate our knowledge and activities, to defeat AMR, and then finally to save life. I think it could be my last slide. Thank you very much for your attention.
Thank you very much, Dr. Vic, for your presentation. So please go ahead with your questions in the chat, or if you want, you can unmute yourself and just speak.
Okay, thank you, Prof.
Warm greetings to my former instructor and Dr. Victoria. Yes, I'm really excited to hear from you. And yes, I'm also in the area of antimicrobial resistance as well. Yes. Yeah, sure. And that's our expectation. Coming back to home and try to assist the researchers as well to for capacity building in this area. But using one of the alternatives that I'm working on currently, which is phages, the use of phages to control antimicrobial resistance. Yeah, that's what I'm working on currently, and the publication will come up very soon. Yes, I know you have worked a lot on medicinal plant. Yeah, I know it's your area of expertise. So my question will be, I know that in your bank in Benin, you may have many medicinal plant. How are you translating those, I should call them plant biodata in order to control AMR? Is it really possible to use chemical means to control antimicrobial resistance?
Thank you very much for your question. Yeah, I think it's possible. You know, in Benin, especially, more than 80% of the population still rely on medicinal plants to treat themselves, even if they have access to pharmacies, they have access to hospital, they still use that traditional medicine to cure diseases. So here, we have what we call the traditherapists, those people who knows the plants. When you say, okay, I suffer from this disease, digestive infection, they know the plants that could help you to solve your problem. So we work those people to collect those plants, you know, and when we collect the plants, we look which of those plants are the most cited, because we need to know where we come from. So when we found the most cited, then we start working, screening those plants. We will look for the toxicity, we will look for the pharmacological effect. Then we will test those plants in vitro on bacteria, and we will create the disease in lab animal models. We will create the disease and cure the disease in the lab. And when we are sure that the plant is working, then we will try to explain how this plant is working on bacteria. For example, recently, I think it was published, we published a work where we found the mechanism of a plant. We call it Caya senegalensis. It's a plant where you can find everywhere. And we find that that plant destroys the negative gram bacteria by destroying the cytoplasmic membrane. You see? So when we know how the plant is working, it's easier to suggest it to the population. So that's how we work. And when we achieve those kind of results, we are convinced that we can use those medicinal plants, at least for a moment, to solve the cure, because, you know, the plants are also giving the chemicals. So after some time, we can have this adverse phenomenon where the bacteria will get known by those plants, and we can face another resistance bacteria. But step after step, we need to move like that. Thank you. I have Can I go ahead, Prof?
Yeah, please.
Thank you.
There are a few questions here. Yeah.
Yeah, I'm looking about medicinal plants. How do you control the quality of the production? Because I suppose that the plants are not safe from antibiotic which spread in the environment. Definitively,
No.
the plants are also facing the same problem, you know, and we look for this, comparing some plants coming from some village region and some plant we have in the city. And generally, when you work with the traditional therapists, they go very, very far from pollution. They go in a village to get their plants. But we look for the antibiotic residues. We have this methodology using ELISA test to look for antibiotic resistance. So we need to see if the amount we found are under than the level required before performing our test. So it's true, the problem is there, and we work with that. How does AMR spread from person like George to other persons? Is it like the COVID spread through aerosol? No, not exactly. You know, AMR is not so visible as a COVID. You know, this morning I was giving a talk, and we were saying, okay, we have COVID today, but AMR will be the next pandemic. Because when I heard some antibiotic, some resistance bacteria with my body, when I come inside the society, when I cough, it's a problem. When I go to toilet, it's a problem. When I go to urinate, it's a problem. Even when we share coffee in the same room, it's a problem because bacteria are in the atmosphere. We don't see them at all. And we are exchanging all those bacteria every time we are trying to talk. So that's how this could be spread. Don't you think this decoction contained? Yes. And I have already answered to the question. Yes, sometimes we have some residues of antibiotic among those detection, decoction. And that's why it's important to secure a way to detect those antibiotic residues. Okay. What are the symptoms to detect AMR? Yeah, the main problem with AMR is that it's silence. You know, the main symptom is that you get ill and we cannot cure your disease. That's all. You have some people, they get infection. We use all the antibiotic that is available and nothing changed. The disease cannot be cured. This is how AMR can be detected. And at this moment, there is nothing to do, unfortunately, because you have already escaped all the way we can, the antimicrobials that could save your life now cannot do anything. Thanks, Doctor. Do you say that AMR is matter of antimicrobial? What kind, which kind of diseases, especially viral and fungal, are currently big matter? No, I don't get this question. Arsene Araba, I will suggest to if you can formulate again the question, because I don't get it very well. Okay, what?
I think here, I think the question is like, what kind of disease is spread through AMR? What kind of disease is the major problem? What,
okay,
like staph or something, you know?
Yeah, we have many even I will talk about Benin, what I know. You know, we have many problems with Pseudomonas. Pseudomonas, where you find some strain of Pseudomonas which are multi-resistant, then we have some problem. And also recently, from what we are doing in the lab, we find some multi-resistant strain of Acinetobacter. And you know, Acinetobacter can be a big health problem, especially among infants in the hospitals. And we found many strains of Acinetobacter recently, and this makes us discuss with the medical doctors so that they know the problem is there, and we can work on changing the habits. At least in Benin, we start something because you cannot have access to antibiotics when you want. You need to show a prescription before they sell antibiotics to you. So the problem is the same with any viruses, any fungi. If we don't pay attention, the antimicrobials that should help us could become a problem for us. That's why when you get fungal infection and they start the treatment, even if you feel better 1 day, 2 days after, you need to follow the treatment. You need to finish the prescription. If not, you will feel better. You say, okay, I'm fine now, and I stop the treatment. And if finally, you will get this fungi will become resistant to the antibiotic, and the next time it will be a problem. You will take your time to spray it, this antimicrobial resistant microbe, and that will be a big problem. What are the mechanisms by which medicinal plant secondary metabolite affect microbial cell? Yeah, I think I talked about this in my talk. You know, it could affect the nucleus, destroy the nucleus. It could work on the cytoplasmic membrane, destroy it or disrupt it, or it can also affect the DNA from the directly of the microbe. So it could disorganize the cell. There is many mechanisms. So when we hear that this plant is effective against this pathogen, our job is to find the mechanism, is to find how it leads to that result. And I think this is how we are working to improve the things. What is the current trend of drug resistance among pathogens? Sorry. What is the current trend of drug resistance among pathogens? I don't know if I understand the question very well. I'm not sure I understand it very well.
So
the current, what do you mean by the pancage?
Yeah, so probably Pankaj probably meant that what kind of AMR is more common, probably.
Oh, okay. Oh, the most
What
one is bacteria. It's bacteria. The most common drug resistance we have is related to bacteria in Benin especially, and we start finding some resistance to tuberculosis. Also, some work noticed that. So it's mostly bacteria. Thank you, Chloe. Thank you all for your comments. Thank you very much. Thank you. Completely agree. And your point about capacity building. Yeah, but how do you think issues like skill gaps in supply chain can be best addressed? Yeah, it's a big problem, you know, Dr. Chloe. It's a big problem. It's true that we need capacity building, but the skills gaps need to be filled because we have many African researchers that are very exceptional. We just need to create the condition for them to learn. You know, when we have some projects, detecting some talents, some young talents, expose them. We need to expose our talents. There we go in maybe in your lab to learn techniques, go to another lab to learn techniques, but you need funds for that. Even if you have the collaborators, you need funds for that stay. And when we will have those techniques in place, then there will not be a problem. And even if the supply chains, you know, in Africa now, we have many, many big factories that are a supplier of reagents. You know, yesterday we were discussing with Inkwaba Biotech. They are doing great, offering some reagents, PCR machines, PCR products directly in Africa. And the delay is not so long. So things are getting better. We need to support it as researcher. We need to work and found collaborative grants. This is the only way we can impact the situation. Because even if for us, we have done our best, we have some good young researchers behind, and we need to promote them. We need to put them together with other researchers in other countries and train them. That's why I'm talking about training the new generation of scientists. Is there any method for AMR detection through NGS in genes with method? Yes, there is a lot of methods. We had a good talk yesterday or the day before with Inkwaba, where they offer very good services using NGS to look for genes with mutation. I can even discuss with Tumpa regarding that issues. Dr. Chloe, thank you very much. Thank you for your comments. Yeah, it's true, but we are there for the change, you know, and I know you are also doing great. So we hope that this will help us to reach a new step. This is my hope also. Thank you. I think I've gone through the question. Okay, are there adequate conditions for effective involvement of upcoming Beninese scientists in diaspora? I used to say, the conditions are related to the researcher himself. If you want the condition to be adequate for you, it will be adequate for you. This is my hope, and this is what I think. The country is there for all of us, but we need to work and take what we need to take in the country. So, Michodi, you are welcome any time you want. The country is yours, and you need to come back, helping us to solve this problem.
Yeah. It's great insight, Victorian. Dr. Vic is a great talk. You know, I have one collaborators from Benin, and recently we submitted one paper on AMR in domestic animals. So how it is spread from, and there is like meta-analysis studies. So yeah, you know, we collaborate as well with Beninese scientists, and they're excellent. Some of them are really good, and and a lot can be done, as you said, through these collaborations.
Yeah.
So probably after this talk, I will send you an email about we can also probably collaborate as well. And if anybody,
Definitely.
yeah, it's a
Definitely.
global problem, and people need to come together to solve this.
Exactly. Exactly. Definitely. Okay, I think I've gone through all the questions. Thank you
Yeah.
very much for your comment. It was very nice moments. I'm very pleased and happy that you appreciate.
Yeah, it was a great talk. Thank you so much. I think Dr. Chloe's talk will be similar to yours, and she'll be talking about AMR as well. So probably you guys can also collaborate in the future. You know,
Definitely.
that will be great.
For sure.
So we'll take a pause maybe for 10 minutes. Maybe after another 15 minutes we'll start. It's about 7:45 now. So we'll start 8 p.m. IST, around 15 minutes. I don't know what's the time in Benin right now.
It's 3:16.
3:16, yeah. So
Yeah.
we'll have next talk at 3:30. I think UK's time is the same. You know, that's it's interesting. It's interesting. You know, the world is really coming together for a reason.
Yeah.
Yeah, it'll be great. It's really nice hearing from you. And
Thank you.
yeah. So let's take a pause for a few minutes, 10-15 minutes, and then we'll come back. Thank you, everyone.
Great.
Stay
Thank you.
tuned. Hello, everyone. Welcome back. So, Dr. Chloe, could you unmute yourself, please?
Hi, Arindam. Thanks
Hello.
very much for inviting me today. It's great to be here.
Thank you for agreeing to present research that is close to many of us, and we have heard an excellent talk from Dr. Victorian, and this will fit nicely with his talk, and that'll be perfect stage for you. And let me take this time and introduce you. Dr. Chloe James is the third speaker in our Champions talk. She's a senior lecturer in Medical Microbiology, School of Science, Engineering and Environment at the University of Salford, United Kingdom. Dr. Chloe's research explores the molecular mechanisms by which bacteria interact with their surroundings. She is particularly interested in how bacteriophages influence the biology of bacteria and in the development and spread of antibiotic resistance. Her work has contributed to a better understanding of antibiotic resistance mechanisms and bacteriophages that integrate into bacterial hosts as prophages. She's active in science communication, public engagement. She runs microbiology-based activities at science festivals to increase public awareness and understanding of AMR. She is interested in how immersive technology and art can be used to enhance learning about microbiology, and developed a network of multidisciplinary collaborations to find new and exciting ways to do this. She is also the co-chair of the Impact and Influence Committee for the Microbiology Society. Little bit about her education. She did her BSc honors undergraduate degree in medical microbiology in the School of Life Sciences at the University of Edinburgh, and did her PhD from School of Biological Sciences at the University of Liverpool. Is that correct?
Yes.
Okay, just checking. And Dr. James also completed several postdoctoral work. This work were funded by NIH, Marie Curie Networks, MRC, Wellcome Trust, and she had the opportunity to work in multiple countries in the US, France, as well as in the United Kingdom. I have already mentioned that Dr. James is a co-chair of Impact and Influence Committee of the Microbiology Society, program lead for the MSc Science Communication and Future Media. These are some of her public engagement and administrative responsibilities. She's also the outreach lead for biomedical science area in the School of Science and Science Engineering and Environment. She has multiple funding with HEIF, which focuses on antimicrobial surface coatings for controlling infection transmission. There is BBSRC funding, which focuses on prophage host interactions, and RSIF funds, which is for launch of next generation sequencing facilities at the University of Salford. And these are just only few. And she has a number of professional membership, including the Microbiology Society, Society for Applied Microbiology, Northwest Microbiology, International Society for Viruses of Microbes. The title of her talk is perfect. Just right after the Victorian's talk, Dr. Vic's talk, the title of her talk is Antimicrobial Resistance of Clinical Staph aureus Isolates from Portal in Uganda, and the Challenges of Research in Low Research Settings, which Dr. Victorian kind of introduced. So it will be a perfect. Please take it from here.
Great. Thanks very much. Thanks for that great introduction. I'm just going to try and share my screen before I carry on. Let's hope this works. And
yes.
Can you see that okay? Great, thank you. Well, happy International Microorganisms Day, everybody. And thank you, Arindam, for organising this event. This is a really great opportunity for us to come together as Microbiology Society champions and talk about some of the stuff that we do. So you've already given me a great introduction, so thank you. This is the Peel building, which is where I work at the University of Salford. L.S. Lowry, if any of you are art lovers, apparently painted from the top window of the building there once upon a time. I want to start just by really talking a little bit about what we do as microbiology champions. We kind of are privileged to have a bit of a remit to help to promote the art and science of microbiology. And the Microbiology Society has been fantastic at supporting us in organising all kinds of exciting activities. What you're looking at here is our immersive room that we built at Media City in Salford for the Manchester Science Festival. And this is a simulated gut that you can walk around and learn about the healthy microbiota. You can also inject bacteriophages into there. You can see how antibiotics affect the microbiota and listen to some of our gene sequencing data that we produced. And so we really enjoy putting these things together, but also some more simple activities where we draw and make art with bacteria on agar plates. So this is just my thank you to the Microbiology Society for supporting things like this. So there's a picture of me up there holding a giant model of a phage, which is a big part of my research. But today I've been asked to talk about our work on AMR, so it's a slightly different tack. And I'm really, really grateful that I'm following on from Victorian, who gave a fantastic overview of the problem of AMR. And there'll be some parallels with what we're talking about. So with Victorian from West Africa and my research, which has been focused in East Africa on a much, much smaller scale though. So because this is International Microorganisms Day, I think I'd like a big shout out for the antibiotics producers. So the penicilliums and the streptomyces and all of the other fungi and bacteria that produce antibiotics, without which we would be very lost. We rely so heavily on antibiotics for treating bacterial infections. As Victorian quite rightly mentioned earlier, antimicrobial resistance is about viruses and fungi and parasites as well. But my research is focusing very much on bacteria that have become resistant to these antibiotics that we rely on so heavily. Victorian had the same information up in his talk, that we have massively been overusing and misusing antibiotics for a very long time. And now we have this catastrophic global threat of antibiotic resistance that if we don't do anything about it, there are projections that 10 million people could die globally by 2050 of resistant infections. And every country has its national strategic plans for tackling AMR, and these are very similar across the globe. There's a real importance and a real emphasis put on improving sanitation and hygiene, and also raising public awareness about AMR and the importance of surveillance. And so I think both Victorian and I have this very close to our hearts, that there are big gaps in the surveillance data, and some of our research is looking to try and redress this gap, but at the same time to help to raise public awareness, but also to work together. Like, people are so important, and people from all kinds of different sectors to come together and work globally, and we do need these international collaborations to make a big difference. So I'm going to talk about our work, which was quite a small scale study, much smaller than Victorian's very impressive body of work. This was a small, very local pilot study in Fort Portal in Uganda. This is a picture taken from a maternity ward at Fort Portal Regional Referral Hospital. Everything looks quite well organized in this picture. The ward can get very, very hectic and very crowded indeed. The staff are very stretched. And they have very poor resources, very limited resources for maintaining hygiene, preventing spread of infection, and effectively treating patients. And so our project brings together microbiologists, molecular biologists, pharmacists, experts in social justice, but also ward staff, particularly at Fort Portal Regional Referral Hospital, to have a holistic view of the problem in this area and to see how we can respond to evidence to improve policies. As Victorian said, the threat of AMR is most keenly felt in lower and middle income country settings. There are very poor resources. Supplies of antibiotics can often be very low and unreliable. There's a huge amount of unregulated antibiotics in circulation. In particular at Fort Portal, the antibiotics are acquired bi-monthly, and by the end of that 2-month period, often key antibiotics have run out. So patients don't always get the full course of antibiotics that they need. They have to go and buy them elsewhere, or are prescribed a kind of a hodgepodge of different antibiotics, which can cause a lot of problems and can promote increase in multi-drug resistance. There are also changes in cultural practices. This is the area, sort of the waiting area outside of the maternity ward at Fort Portal Regional Referral Hospital. And you can see that there are a lot of people here, and they're here all the time. These are attendants that will travel very far with their relatives that need treatment at the hospital. And we'll stay here, we'll sleep here, we'll cook here, we'll often cook in the wards, sleep on the floor of the wards. And so the issue that Victorian was talking about of One Health, of bacteria being spread via healthcare workers, but also attendants, and then going back to their homes, and that the circulation of antimicrobial resistance is very difficult to get a hold on. So our work is just focusing on clinical isolates at the moment, but we do have interest in looking at isolates that are carried on healthy skin and also in animals. So the main thing that we're addressing in this study are the gaps in surveillance, but also addressing how people interact with each other in hospitals to try and improve antimicrobial stewardship so that they can be used more appropriately. So Uganda has a national action plan that was drawn up in 2018. Some of the highlights from that, just to reiterate the problem in Uganda. So Uganda is here on the east of Africa, just above Lake Victoria, right on the equator. It's a very small country, and suffers a lot of the same problems that Victorian was talking about from West Africa. Over 37% of hospital admissions are due to bacterial infections in Uganda. And if you look at the number of deaths per, that should be per 100,000, caused by infectious diseases, it's much higher in Uganda than in developed countries. The National Action Plan has highlighted several priorities, including detection of AMR and surveillance of AMR pathogens, as well as improving IPC and public awareness about AMR. One of the big problems, so the National Action Plan has focused on a number of hospitals throughout Uganda, trying to equip them with the resources that they need for antimicrobial susceptibility testing. But a lot of the data that exists so far is from densely populated areas in the bigger cities, yet 76% of the population in Uganda live in remote areas. And so there's not much information from those more remote areas, and that needs to be redressed. So our study is based in Fort Portal, which is to the west of the country. This is about a 6-hour drive from Kampala, the capital. And its location is quite important because it's right by, this is the Rwenzori Mountains here on the border of the country, and on the other side is the Democratic Republic of Congo. So a number of years ago, Fort Portal was highlighted as a possible entry point for Ebola, particularly around 2014 and 15 when the first biggest outbreak was reported. And so the hospital is quite well resourced compared to other hospitals in the country, and they do have a microbiology lab there that at the moment is run quite well with the help of the Infectious Diseases Institute at Uganda. But still in Fort Portal, rates of maternal mortality are huge, despite massive investment in the area. They are still disproportionately high, even compared to the rest of the country. Antibiotic supplies are very poor, but very poorly regulated, and there's very little data on AMR from the area. So our pilot study started in 2017. In between that time, so from in between then and now, the lab facilities have been massively improved, and the systems in the hospital have also been massively improved, partly because of the involvement of the project that I've been a very small part of. So we collected 41 isolates of Staphylococcus aureus. I say we did, the ward staff did at the hospital, and the microbiology lab there processed, identified the samples using API strips, and performed distribution assays to profile the resistance profiles of those isolates. And so that was done by the microbiologists at the hospital. But then we shared those isolates, or they shared those isolates with us, and we extracted their DNA, and we performed whole genome sequencing to look to see how much the genetic and phenotypic data agreed. So this slide's a bit data heavy. All of the data is on this slide, I'm afraid, but don't worry, I'll talk you through it. So what you're looking at here, as we published this in Microbiology at the beginning of this year. So we sequenced the 42 isolates. There were some isolates that weren't Staph aureus, they were Hemolyticus, but it was a great testament to the lab in the hospital that their identification techniques were pretty strong. So the circles that you're looking at show whether or not the isolate was susceptible in white or resistant in black to the antibiotic. So this is the phenotypic data. And what you can see from this is that there is quite a lot of resistance. In fact, 73% of isolates showed resistance to at least one antibiotic. And 29% of isolates were multi-drug resistant, and some of them were approaching pan-drug resistance. So you can see some of them here are showing an awful lot of resistance. If you look at the black squares, this is where the equivalent resistance gene was present in the whole genome sequence. So you can see that there are some genes that are incredibly prevalent in the population, such as dihydrofolate reductase genes that encode resistance to trimethoprim, and also some beta-lactamase genes as well. So we compared these profiles, and so in this graph here, this is showing the solid color is the antibiotic phenotypic resistance, and then the blank column next to it is the equivalent gene. So here we've got ciprofloxacin resistance at about 30%, and the presence of mutations that confer resistance to ciprofloxacin is quite similar. So in a lot of cases, we can explain a lot of the resistance by identifying the equivalent resistance genes in the genome. But this wasn't always the case. So a couple of points that I want to highlight, first of all, is with cefoxitin. So cefoxitin, is a third generation cephalosporin, and when isolates are resistant to cefoxitin, this is an indicator that they are an MRSA. So this is very commonly used to report MRSA, but the gene presence didn't really match up with the phenotypic resistance. But this is because we're not looking at the same number of isolates. If you have a look back at the phenotypic data for the individual isolates, you can see that there are a lot of circles missing for cefoxitin. And this is because the resources were very poor at the hospital and they ran out of cefoxitin discs. And this happens fairly regularly. And so what is quite worrying is that there are some instances at the bottom here where the assay was not performed because there were no discs, and these isolates did in fact have the MEK gene indicating that they were MRSA. So the poor resources are meaning that the hospital is missing important detection of MRSA that would affect the treatment because they would need to change their empirical treatment to another antibiotic in response to this. And so this was something, this sort of a real bit of information that we could feed back to the hospital to try and improve their procurement of antibiotic discs, to improve their detection systems. The other thing that I wanted to point out is a bit of a good news story that we found no resistance to clindamycin at all. 100% of the isolates were susceptible. But we did find erythromycin resistance. So these two antibiotic classes are related. They have similar targets of action. And we did find about 30% of the isolates had resistance genes, the ribosome protection genes and efflux pump genes that confer resistance to both of these antibiotics. So by just looking at the phenotypic data, it looks like the isolates are sensitive to clindamycin. But what the genetic data is telling us is that there might be inducible resistance to clindamycin if erythromycin is used. And that there are tests that can be done to identify this. So this is really just a watch point for the hospital. At the moment, they don't use clindamycin, but just because there's clindamycin, that the isolates look so sensitive, they may well have the resistance genes and may show resistance very, very quickly if clindamycin were to be used. So these are two bits of information that are very local to the hospital, but I'm hoping that this data can show you the power of local evidence to improve hospital systems, and also it's really useful in training as well. Using the whole genome data, we did a phylogenetic analysis, and we found that there were a lot of clonal complex 152. So a lot of them were very, very similar. There was no evidence here that this was clonal, so no evidence that this was a one strain that was spreading throughout the hospital. And this is a common strain across sub-Saharan Africa, so this wasn't surprising, but it's nice to be able to show this data. So in parallel to the microbiology and the molecular biology that was being done, the social scientists were performing action research at the hospital. And this is through Professor Louise Akers, who's been working in Uganda for about 15 years now. And it's her that invited us to come and be involved in the project. And she's been working for a long time with sustainable volunteers. And so this is bilateral movement of Ugandans and people from the UK to volunteer in sort of clinical positions to learn about different systems. And so during that time, they've done a lot of analysis of facility data at Fort Portal Regional Referral Hospital with qualitative interviews and focus groups with not just the clinical staff, but the lab staff, the pharmacists, the waste management team, and the technicians to try and get a holistic view of what's happening at the hospital. They have a big focus on maternity because maternal death rates are so high. But they found that the maternity ward is by far the highest user of antibiotics at the hospital, and that midwives play a critical role in the prescribing of antibiotics, but they have quite a poor understanding of AMR. So there's a real need there for increased training in AMR. And so we were able to use our data as part of training to really empower people in knowing that they're looking at information from their actual hospital. And so the research found that task shifting was actually really a very useful thing. And so nurses and midwives and clinicians would learn about what the cleaners had to do, also what the lab techs had to do, and what the pharmacists had to do. And this really improved engagement in sending samples to the lab and understanding what the results meant when they came back from the lab. And it also helped the pharmacists to develop the first antibiogram for the hospital so that they could better inform the antibiotics that they were going to buy for the next period. So together, this work was really impactful. The multidisciplinary approach enabled a more holistic approach to systems change to tackle AMR. So far we have recorded a sustained improvements in antimicrobial stewardship at the hospital. And the evidence base from that antimicrobial susceptibility data, as well as the whole genome data, really helped to to galvanize people's thoughts about antimicrobial stewardship. So, as well as the work that we did at the hospital, as I said at the beginning, we do quite a lot of work with different ways of conveying information about AMR. So we've developed a virtual reality experience that talks about bacterial communities and bacterial biofilms and how they respond to antibiotics. And it has strong messages about the importance of taking the correct antibiotics and completing the correct course. So we had a focus group with this, with the bioinformatics students at Makerere University. And I mean, their knowledge of AMR is fantastic already, but they really enjoyed this experience. This is Daniel, who's the lead of the microbiology lab at Fort Portal Regional Referral Hospital. And this is Joe, who is a pharmacist at the hospital as well. And they were absolutely crucial in this project. And it was really useful to look at the virtual reality experience with them, and they really felt that this experience helps to enhance conversations about AMR and helps people to look at things in different ways depending on what discipline they're from. Other things that we've done are placements. So we have students from Salford and across the world, to be honest, that can spend between 3 and 12 months in Fort Portal working with clinical professionals. So this is the blood transfusion team that go out into the local area and collect blood, but we also had projects of collecting isolates and looking at resistance profiles from isolates out in the community. But also working in clinical diagnostics labs, diagnosing HIV and syphilis and malaria and TB. And this was a really important learning experience about how healthcare is very, very different in under-resourced settings. We also travelled around local pharmacies to see what antibiotics were being distributed and looking to see whether the right antibiotics were being prescribed for the right things. So lots of different snippets of work that all come together and build a big picture. I was really glad that Victoria made the point about capacity building, and that's why I asked a question about it earlier on. Because I do feel very strongly that there are fantastic networks of passionate researchers and people with very good bioinformatics skills and microbiology skills across sub-Saharan Africa. And we performed our whole genome sequencing at Salford, but this is Moses who came, so he works at Makerere University. So he came with Gavin, our PhD student, and performed the sequencing at Salford. This is him getting involved in one of our activities at the Manchester Science and Industry Museum. But then we also repeated that sequencing back at Makerere because they have their own MiSeq sequencer there. But there came across a lot of challenges around paperwork for moving strains and getting various approvals and things like that. But also in getting hold of the reagents. So reagents for sequencing cost about 4 times more if you buy them in Uganda compared to if you buy them in the UK. And I think this is something that really needs to change. So this is Poppy and Judy. They're working on a different project with nanopore sequencing, and I think there's a real some real promise in nanopore sequencing, including surveillance of COVID as well, but that's a completely separate issue. But I do think that this is a conversation that is really important for improving capacity building in low resource settings. So lessons learned from the project, talking to others in different areas really can build strong research collaborations and help to overcome challenges. I think this project would not have worked if it hadn't been equally, had equal efforts from people from across Uganda on all different sectors. Action research can be messy, so this is the culmination of about 3 years work of lots of little bits and bobs, and sometimes things can go very, very slowly, getting permissions and getting facilities right for doing work. But then sometimes we found that we were able to produce work that had a lot of immediate impact, which is also very rewarding. So working across countries and in different disciplines really helps to widen your perspective and strengthen the impact of the project. So that's all I wanted to say, apart from just to thank all of the many people that were involved in this project. I'm not going to go through everybody in person. I just wanted to particularly highlight Gavin because he did a lot of the groundwork, and he had just submitted his PhD thesis this week, detailing this data and a lot of related data around that. And Louise, who really started off the project about 15 years ago, and she's a professor of social justice and has done a lot of work with empowering people working together in clinical fields. So I think I will leave it there. I will also mention David Katiti and Henry Kajambula at Makerere University, who were instrumental in actually getting us to be allowed to do this work and to get everything running smoothly. So thank you very much. I'm very happy to answer any questions.
Thank you very much for the excellent talk, Dr. Chloe. I think we have some questions. So, yeah, it's in the chat.
Ah, okay. Okay. So very serious problems. The images are related to the difficulty of monitoring antibiotic resistance in an environment like this. The poor are giving us a bad tour. Many efforts have been made, only action must be taken. Thanks for the presentation. Okay, so thank you. Yeah, so I agree. I think that a lot of countries have developed very good and very promising strategic national action plans that have very clear and definite priorities, focusing particularly on One Health, and we really do recognise the huge problem. Of course, as Victorian said, this really needs big multinational involvement to sort of get a better feel for the resistance in areas that haven't really been looked at yet. I think that during the project, I've noticed that across all sectors, there's a lot of passion about AMR, and there's a lot of conversations about AMR, but we desperately need more funding to be able to get on top of the issue. Okay, I'm asking if it's the transmission of the multi-drug resistant or XDR tuberculosis in an environment like this? Yeah, so extremely drug resistant tuberculosis is definitely a huge problem, and it's definitely growing. It's not my area. I know that Makerere University have a fantastic TB lab. Fort Portal Regional Referral Hospital has a separate ward for TB, and it's dealt with quite differently. I know that they reserve use of rifampicin only for TB cases. Definitely a big problem that is on the rise, but I don't have any specific information about Mycobacterium tuberculosis data. But yeah, absolutely a huge problem, and something that's not going to go away anytime soon. So clindamycin is often used in local hospitals. If not, can you explain these results? Okay, so at Fort Portal, they don't use clindamycin. So I've seen all of their procurement lists, and they don't have it on their list. But that's not to say it isn't used. Certainly in Mbale and Malago Hospital in Kampala, they do use clindamycin. And clindamycin resistance has been reported in other areas in Uganda. And this, I think, is why it's quite important to have this high granularity, so looking at very local data, because I think when you have national or international guidelines about the use of certain antibiotics, it doesn't always necessarily apply everywhere. So in Fort Portal, there was 100% susceptibility, but it would be very, very dangerous to suggest that it was okay to use clindamycin, because there's definitely resistance that has been reported elsewhere. There is a test that you can do. It's a variation of the dysdiffusion assay called the D-test, where you put an erythromycin disc and a clindamycin disc close together on your agar plate, and it will tell you if there's inducible resistance or not. So what I would like to do, we've got another project student going out in January, and I'd like him to do some work with the D-tests there to see whether there's inducible resistance at the hospital or not. But this is another issue that I think, particularly in under-resourced settings, the availability of certain antibiotics just isn't there. And so I think that there's going to be different resistance profiles because of the difference in use of different antibiotics, which is why I think this quite intense surveillance is really quite important to get a handle on what's going on. I hope that answered the question. Thanks, Victoria. And yeah, so I agree, very complimentary work. And yeah, we definitely need to chat more. Yeah, yes, we'll watch this space. Oh, okay, so this question, is it also possible for Benin to benefit from these placements? It's a really good question. So we work with, so Louise also heads a charity called Knowledge for Change, and Knowledge for Change has been mostly based in Uganda, but has also had interactions with other sub-Saharan African countries, and I think also potentially in Indonesia as well. So there definitely is a desire to work with other countries. So I can ask the question and I can get back to you on that. But even if, I mean, obviously Knowledge for Change is quite a small charity, but it's a model that could be rolled out elsewhere as well. And yes, I will put you in touch with Louise, and we'll keep talking. So the bridge building activities. Okay, so this is talking about the sequencing and the virtual reality and things like that. So a lot of the activities we do, Salford partners with Manchester Science Festival, and so we do a lot of science festival-y things, but lots of the activities that we develop for that are applicable all over the world. The virtual reality, we are hoping to, the next stage of that is on its way, and we're hoping to develop it so that people can download it onto their own phones and use it with cardboard headsets, so that would be just freely available for anybody to download. And I'm really interested in maybe co-developing with somebody educational tools around that. The experience itself is fairly basic. It's not perfect, but it's a brilliant trigger for conversations. And so Daniel Kimbombo in the Fort Portal was saying how it could be really useful for tutorials with at universities, but also with other clinical professionals about their understanding of AMR. And so I'm very keen for that to be distributed as widely as possible and for anybody to use it. We have a website with more details of that, so I can share that with you. So yeah, definitely send me an email and we'll see what we can do.
Yes,
Okay.
Excuse us, Dr. Chloe.
Sorry, say that again.
Yes, may I ask my questions?
Yes, absolutely, please do.
Thanks for your fantastic talk. I know also you are an expert in bacteriophages research, and
yes.
I would like to hear from you on this aspect of the use of bacteriophages to tackle AMR, especially what do you think that bacteriophages can be used in human one day in Africa? Because I know in Poland, in Greece, in USA, people are using it for compassionate purpose. But coming to Africa, you have been in Uganda. So do you think that this can be implemented when it's come to human beings?
That's a massive question. As I guess I should start by saying my bacteriophage research is on temperate bacteriophages, and these are prophages that integrate into bacterial genomes and carry virulence factors, and they have a very integrate interaction. And those particular bacteriophages are not recommended for phage therapy at all. But obviously, I've been to a lot of phage conferences, and so I'm sort of up to date with what's going on with phage therapy. I think that phage therapy has absolutely fantastic potential, and there is such a huge body of evidence of individual cases where phage therapy has been used, as you say, in compassionate cases, and has been very effective. The big problem with it is that phages are so specific. So the reason that antibiotics are used so widely and are so effective is that you can use a small number of antibiotics that are effective against a wide range of bacterial species and strains. And obviously that has its problems using broad spectrum antibiotics, which has induced resistance. With bacteriophages, they are highly specific. So you can have a highly specific cocktail of bacteriophages that will be effective for a very small range of strains, which on the face of it is fantastic because you can destroy the bacteria that are causing the infection without affecting the healthy microbiota around it. The issue with it is that it's quite labor intensive to develop those bacteriophages that will be effective across a broad range of cases. So the Tbilisi Institute in Georgia, in the former USSR, has been doing this for decades, and they have huge banks of phages that they can train up for a particular case. The similar institutions are now coming up in the US. The Baylor Institute does the same thing in the US. They train bacteriophages and evolve them so that they can be used to treat a particular outbreak in a hospital, for example. And they've been shown to be really effective. The problem is that we still don't have a huge amount of massive double-blind, placebo-controlled clinical trials that show that the bacteriophages can be rolled out for broad use globally. And there are some big trials that are coming out now, and phages look generally safe, which is fantastic, but it's quite labor intensive to get a phage cocktail that's going to be effective, and it will only be effective for a certain period of time. The treatment needs to constantly evolve. And it can constantly evolve because phages can evolve, and there's this arms race between phages and bacteriophages. Sorry, between bacteria and bacteriophages. But I think there needs to be a change in how drugs are regulated to be able to approve phage treatments. And it's something that I think has fantastic potential, and I'm really happy that efforts in bacteriophage research are increasing more and more people are looking at it. But I think there's still quite a lot of work that needs to be done before we can happily say that phages are absolutely the answer to AMR on a global scale in humans. And I hate that I sound like I'm being a bit negative there, because I think phage therapy is fantastic. But I think that we're not quite there yet. Does that answer your question, Newton?
Yes, it's okay, Doc. Another question is that from my experience, this will be a plea toward the French-speaking country, especially in Africa. I have come to realize that most of the researchers from West, from UK, USA, they collaborate more with Ugandan, East African countries. But when it comes to West Africa as well, they collaborate with Ghana and Nigeria. Yeah, sure. In Burkina, I think there are some researchers from USA as well there. But I want to bring the cases of maybe Benin and maybe Niger as well, and Cote d'Ivoire, yeah, more French people there. Why you don't really look at those key countries? I'm taking the case of Benin Republic. Why your attention is not focused on those on this country compared to Ghana or Nigeria?
It's a very good question. I don't feel equipped to answer it. My very short answer is je sais pas. I guess language barriers could be one element. I did work for a couple of years in Marseille, and I do love speaking French, so I would love the chance to collaborate with French speakers. But it's a good question, like, why do people collaborate with certain countries? And I don't know the answer. I don't know whether there are certain agreements with different countries' governments, but I'm afraid I'm not equipped to answer that. But it's a really good point. I would love to work with Benin.
There is another question about, do you think that lack of in vitro diagnostic manufacturers in Africa is also a factor for AMR increase? Again, what you said, I think earlier, about low resource settings, lack of antibiotic tests, diagnostic manufacturer, lack of it.
Yes, I mean, I don't know any information about that, but yes, absolutely. I mean, more accurate diagnostic tests is crucial in preserving use of antibiotics when they're needed. Again, it comes back to the need for more funding, for more diagnostic tests. I think there's a big problem. I think there needs to be a holistic view at how the funds are best spent, because there are some fantastic technologies, but the technologies need to be shared along with skills and infrastructure and big pieces of kit that can be maintained. One of the big problems, I think, in Fort Portal is that sometimes people will donate a big machine or a big bit of kit, but there isn't the funding for that bit of kit to be maintained long term. And so changes need to be sustainable. And I don't know. I don't know what the answer is, but I think there needs to be sustained conversations about, yes, improving supplies of antibiotics and education about antibiotics and antibiotic testing. But yes, you're right, also diagnostic testing as well, so that antibiotics are only used when needed.
One more question regarding the scenario of AMR among ocular isolates. Do you have any update?
I don't have any knowledge of that. So the isolates that we worked with were from mostly from wounds, in fact, wounds, urinary tract infections, and I think there was the odd ear infection. I don't have any information about ocular isolates, I'm afraid. So Arindam, I think you've frozen, or I have frozen. I don't know. Somebody's frozen. I apologize. Je suis désolé.
Yes, it's je suis désolé. You use it to apologize.
Merci. Okay, well, if there are no more questions, thank you very much. I've really enjoyed coming, and it's been really nice to have a West East Africa conversation this afternoon. Okay, so I think Arindam may have crashed, but thank you all for coming. And I would just like to say again, thank you so much to Arindam for organising this event today. And hopefully it will trigger some new collaborations in the future. So thank you very much. Bye-bye.
I think Prof. Arindam left, maybe due to network, I'm sure, because he's not online no more. So, Chloe, if you agree, I think we went through all the talks. I don't know, because I don't know how I became the administrator of the talk session. So I think we will conclude the session. But together we take this responsibility together, and let Prof. Arindam know it when he will come back. So thank you very much. Thanks to all the participants worldwide. We had a very, very, very good, incredible session. It was a pleasure to be among you. Thank you, and we will organize more events like that. So, Dr. Chloe, if you want to give one word, then we will conclude like that. Thank you.
Thank you, Victoria, and merci. I really enjoyed the afternoon, and yeah, really fruitful discussions. I really hope Arindam's okay. But again, happy International Microorganisms Day to everybody, and please do enjoy your evening.
Thank you very much. Bye-bye. Au revoir à tout le monde. Merci beaucoup.
Au revoir.
Yeah.